Okay, so just real quick on our regulatory drivers. I think most of you guys are pretty familiar with this, but Endangered Species Act of 1973 required federal agencies to conduct actions in a way that didn't adversely impact the future survival of endangered species on the land in which they were operating. This applies to the Army. In our case, it applies to any other organization such as Department of Transportation or anybody like that undertaking an action that involves our taxpayers' money. Lots of Hawaiian species were listed in the 80s, so everybody knows endangered species capital of the world, many endangered species. Um, After the species were listed, there was a time when Department of Army and other DoD branches were getting educated about sort of what the Endangered Species Act meant and what species were on their lands and impacted by their training activities. Our program really started in the 90s. The first field staff was hired in 1995, and that included Kapua and a couple other people. So we went through a long, drawn out consultation process with the U.S. Fish and Wildlife Service, who's the ones that oversee the actions undertaken by federal agencies and their impacts on endangered species. So after many years of meeting and planning and developing strategies, we today are responsible for looking at the impacts of 51 endangered plant species. We use a term called stabilization, which isn't exactly recovery. It's a step below recovery, but it's meant to make sure that there isn't any further degradation to the status of the species in the wild due to Army training activities. Lots of those things that we end up doing are controlling threats. So lots of threat control I'll talk about below. There's one endangered bird that we work with, the Oahu elapaio. So we do quite a bit of threat monitoring or threat management during the breeding season and monitoring populations as well. There's one endangered tree snail species that we're actively managing right now in the Waianae Mountains, Acatella mustelina. There's some other interesting arthropod species that we're really just beginning to understand how to manage and monitor. Those include picture wing flies, damselflies, yellow-faced bees, and in addition, we are working with the Hawaiian hoary bat. Okay, so threat control, I think this isn't anything new to anyone. So there's quite a bit of fire is the biggest threat. So fire threat mitigation is a big part of the approach with training restrictions, soldier education, and vegetation invasive species control to reduce fire threats. One of the basic tenets of all conservation in Hawaii is fencing. We do lots of ungulate fencing to keep pigs and goats out of our management units where we're focusing our management. We also do some pretty specialized fencing, like our snail enclosures, which are made to keep out additional threats that I'll talk about later. Threat control, a huge amount of what we do is invasive species management, invasive weed control. Bread and butter of conservation for us. We really need to get out there and control those invasive plants so that we can have good habitats for our endangered plants and animals. We also do extensive small vertebrate control, rat control on an ecosystem scale in a number of areas with a variety of tools. We're involved in invertebrate threat control. We do quite a bit of slug control in and amongst lots of our endangered plants that are susceptible to their impacts. We're involved in the coconut rhinoceros beetle efforts, and we're also actively looking at ants as a threat. Coupled with that, we do an extensive amount of native forest restoration to provide habitat for our endangered plants and animals. So this is an interesting slide. It kind of just gives you a real quick breakdown of what life is like here at ANRP. So our field crews in general do about a third of their work controlling invasive plants. Other than that, we spent about a third of our time as well in the office. That covers all of our office side, including staff there in the office 100% time doing database and GIS actions, as well as all of our planning actions. We spend about 8 to 10% of our time on our rare plants, working on those. We have extensive greenhouses. We do lots of propagation, both of endangered plants and common plants. We also look at some interesting weed issues in our greenhouse, about 7% of our time. Most of our ungulate fences are built, which is great, and that's why that labor is down to 4%. Mostly what we're doing now on our fences is just maintenance, making sure that they're staying tight. We're keeping the animals out. We spend about 3% of our time right now on endangered tree snails and flies, small vertebrate control, ants, the same 3%, slugs, other threats, coconut rhinoceros beetles and ants, 2%. Vegetation monitoring, we do extensive vegetation monitoring across most of our heavily used management units, about 2% of the time. 2% of the time on outreach activities, that includes Army outreach as well as public education, school groups, everything. 2% on Elepaio management, and luckily we don't have to do that much ungulate control these days. We're only about 1%, making sure that our enclosures are staying clear of invasive ungulates. Okay, I don't expect you guys to really get this whole chart, but we just wanted to give you an idea of kind of what our organization chart looks like. So we come under the Office of the Vice President for Research and Innovation. Our PI is Vasilis Sirmos. We're very happy to have him. He's a great PI, really strong advocate for us in all things that we need him. We have an excellent administrative officer there, Tracy Nakagawa and Melissa Arakawa are great administrators that work with us. Below that, the program sort of divides into two forks. I'm on the natural resource field side. I supervise the supervisors of 4 field teams. 3 of these field teams do all of the conservation work in our management units, from killing the weeds to planting the plants to getting the slugs to controlling the rats. Everything is done by them. And then we have 1 specialized team that does vegetation, restoration, and control. The weed control and the restoration efforts are so intense. We have our own team that just specializes just in that to make sure that we can make some of those goals. We have an outreach specialist, and we have a safety and logistics guy at the base yard that helps us with all of the access and vehicles and all that sort of stuff. Administrative, we have a small administrative staff on project that handles all administrative stuff. We have a pretty good raft of student hires. Great to have them. They're more or less integrated with the field crews, working out in the field as much as possible. A couple of temp hires, intermittent hires. And then on Jane's side of the organization chart, it's divided into 3 different program areas. Animal program, our coordinator is Tyler Bogardus. Rare plant program, Tim Chambers. Vegetation is Melissa Valdez. And those program coordinators work with Jane and me and the teams to design all of the actions that we're undertaking in those areas. We have a monitoring program biologist that covers all facets of biological monitoring, from vegetation to slugs and rats and everything. And then we have a great team that handles all of our information, our databasing, and our GIS. So that's our program structure in a nutshell. We've got 57 positions total. It's great to have that many people dedicated to doing the conservation. We got a lot of great people. Okay, so just a quick snapshot again. This is island of Oahu, and these are the areas that Army trains in currently. Both Waianaes from out Dillingham, Makua, across to Schofield, Lihue, Kalawa areas, in the Ko'olaus, Kahuku, all the way down to Kaukonahua, Schofield, Waikana area. Yeah, these are not the only areas that we work in. We work extensively in state lands. We work on water supply in Pakaha. We work on private lands and other places. Wherever there's a good conservation opportunity, we try to get there and do the work for the species that we have under our jurisdiction. Okay, so the reason why we're coming and talking to you today is because we're really excited to kick off this new partnership with UH. We're working specifically with NREM, and as I mentioned, we've got Clay working with us as well. We put out a call, Melissa put it out, Creighton helped a lot. It's been a really good effort to fund 2 GA ships this year, and that's something that's in our budget in more or less a permanent way. We're looking forward to hopefully funding 2 GA ships every semester for the next few years at least. So let's see. The important things here are that, you know, it's your basic GA ship at UH. Some of that is down here. We're providing support for the GA ships beyond that. You know, we're a great resource as far as getting people into areas, supplying resources for them to do the work, possibly working in our greenhouses and other things like that. So this is coming up quick. They're due the 31st of this month. So that's why we're the first seminar. Melissa emailed us and said, you guys should try to get in, and it would be great if you could do the first one, because that would be before the due date. So we wanted to do this before that and get this out so that people could consider it as an opportunity. All right. Okay, so that's kind of the first part of the presentation. Got through that, just a brief introduction and a quick talk about why we're here today. Now we've sort of divided our research topics into these 6 areas. We're going to go through each one of these areas and present to you guys a few questions that we have in each area. Know that this list is in no way complete. It's a great starting point for thinking about how we can do things to improve our conservation actions on Oahu. There's probably a lot of great topics that we haven't even thought of that aren't on this list, and that isn't because we're not interested. That's just because we didn't get that question asked yet. So to step back a little bit from that, we've always, fortunately, collaborated with UH. You can see UH is by far the most cited the most on this table here, and that's for good reason. But we've also worked with a lot of other affiliations. But we've been doing research with different people at UH on a variety of topics for many, many years. So yeah, you can see some of the examples here. Not going to go extensively into this, but yeah, we've worked with Paul Krushelnicki over many years on some really interesting topics that have really helped us understand some of the things that we really are just beginning to understand about ants and about flies, about the impacts of rats. We've done some really interesting plant stuff over the years, some really informative and definitely formative in terms of our management plans, rare plant genetics with Morden at UH, some of the work that Lauren has done, Weisenberger as well, just have shaped our management and continue to shape what we do today in the field. Yeah, so forth and so on. I'm not going to take too much more time on that. Okay, so let's jump into the different 6 areas. So we're going to talk about ecosystem and restoration research efforts. So you can see A24 over here, our principal kind of rat control approach. Here's a victim of our high-speed New Zealand trap underneath. This is one of our great employees killing a devilweed in Kahuku. So, you know, there's all sorts of facets of ecosystem restoration, lots and lots of stuff to talk about. How do we best monitor rare resources in terms of their impact or in terms of their response to threat control? So some of the great examples of this are like in New Zealand, right? They've been doing this kind of stuff down there for much longer than we've been doing it. And they know that they have to control stoats down to 5% activity levels in the areas where they want to get kiwi to actually produce babies, right? So they know like what percentage they have to get their predator levels to produce offspring in the bird that they're trying to conserve, right? Which is just perfect. They've linked their threat control to the resource response. We are just in the very beginning steps of being able to do that. We'd love to be able to say, if we want to get, you know, this much Cyanea superba fruit, then our rat tracking tunnels need to be reading 10% or less, right? We don't really know what that number is. It's just a guess. It'd be great to be able to say, even like for a weed species, like say you've got, you know, rubus thimbleberry in the understory and it's choking everything out and all the native plants are having a really hard time germinating or, you know, reaching maturity through it, something like that. It would be great to say, if we want, you know, native plants to survive, we need rubus to be at 10% cover or 50% cover or something like that. That could inform our weed control strategies. So those are kind of some examples of that first one. Okay, the next one is an interesting one, and it's quite a bit more specific. So this one is in relation to Hawaii provisional seed zone maps that Loomis and Keir developed. I'm not sure how much familiarity you guys have with that, but it's a super important tool that informs us about where we can use seeds. So basically, the island is divided up into some zones. Windward, leeward, some real basic kind of designations there. And those tell us that, hey, you know what, if you collected ohia seeds from this mountain, from this ridge over here, you can use those ohia seeds to do reforestation across this area, but, you know, maybe you shouldn't use them on the other side of the island. So that's what it does. But it's just a first cut. And really what needs to be done as a second step on this is some common garden experience need to be used to look at, okay, if you do use those seeds in this kind of an environment, do they do as well as we think they should? Or are they so specifically adapted to where they are that that seed zone is way too big? So there's some interesting questions there that would really help us inform our restoration approaches. Okay, the last one here is, let's see, increasing efficacy of monitoring to inform adaptive management strategies to increase rare species. So let's see. This one, although it's not necessarily a real specific question, we'd love to design more monitoring strategies that would help inform our restoration program. So, how much guava do you need to take out of a management unit before you get a positive, gain in a common native species, or such questions like that? More sort of broad stroke questions.
Yeah, so just I think with these kinds of first three questions, just to kind of to build on what Joby was talking about, is we always need kind of new methods of figuring out, especially with that last question, new methods of figuring out if our work is having the desired outcome, and if there are ways we can improve our efficiency or improve our management actions so that we are kind of supporting meeting our goals in the best possible way. So there's, I think that third question in particular is really broad, and it kind of speaks to how there's a lot of room for a lot of questions. Like when we sit around the office and brainstorm, we come up with way more questions than we would ever have time to approach. But yeah, so I think in part, we definitely are open to suggestions from any potential researchers about potential topics. But I think that the provisional seed zone map one is a very specific kind of topic, and speaks to like how much like a specific need that we have. So yeah, you'll see with all of these questions, there's kind of a variety of highly specific topics and pretty broad topics. Okay, I see one in the chat now about climate change for number 2. And yeah, that's a great question. And it's, I think, a really important one. And it's one we're thinking about for both common native species and for rare taxa. So we started thinking about it a little with rare taxa with some work that Lucas Fortini has done, kind of modeling where the climate envelopes of conditions conducive to a particular plant will be, say, in 30 years. But of course, that's only one part of it. We need to know where the habitat of these species is going to be in 30 years. So I think that would be an additional element with the provisional seed zone map, right? To incorporate that climate change. That'd be a great way to develop that. Okay, I see another one in there, a field study with a lab study for questions 1 and 3. Yeah, so I think for sure, I think those could be combined, field and lab. I think with like a lot of the challenges with monitoring rare resource response to threat control measures is sometimes we can do a specific study that looks at the impact of one particular threat on one particular species, but it's challenging to know whether there are other variables at play in other types of ecosystems or with other species. So I think that's where like having really controlled things, maybe in the lab or the field, can be really informative. I do think one of our big needs is in the field, like how do we tackle measuring success when there are multiple variables at play and multiple different potential metrics for success. So I think there's could be some good discussions about how to do that. But yeah, I think that's one of our biggest needs, and is figuring out how to gauge success in the landscape in response to active management work.
Jane, I'm just going to follow up on that to say, maybe in this case, instead of saying lab, is saying greenhouse more appropriate? greenhouse testing of things. And then secondly, I know Joby briefly spoke to it earlier, but maybe if you can just reiterate the types of access and resources that a researcher would typically think about to try to test these things in the field. You guys are actually amazing about providing, even if you're not directly providing funding to a researcher. Can you just speak a little bit more to that? Because I think that also helps to answer this person's question.
Yeah, that's a good point. Lab and greenhouse are both are both right, more of a controlled out of the field settings. But yeah, so in terms of like the sites that we work at, so in general for research projects, we do ask researchers to kind of go through a research agreement process with us. And what that does is it helps to lay out clearly like what expectations are on each side in terms of support we can offer. So whether that's access to field sites, sometimes it includes transport, sometimes not. It kind of depends on the needs, but that's one thing up for discussion. Sometimes that transport is just a vehicle, and sometimes it's a helicopter for some of our more remote sites. So where we can, we try to see if our with the research topic and our existing management work, if we can kind of do those things together. Another thing would be like plant material, or I guess that's an easy first example. But like if we're interested in, say, one of these common garden experience, we might be able to help supply some seed or propagules for it, or help to facilitate collection of those. The one thing that we can't really do is we can't provide permission to work on somebody else's land. So say if one of the sites is on state land where we do a bunch of management work, then there would still have to be a state permit that the researcher would have to go through, because we can't grant that. So I'm sure I'm missing some stuff. Joby, you want to jump in?
I think maybe one of the biggest ones to add to that, Melissa, and something that Clay was talking about, is that we've got so much data and we've got so much history with the lands and the species that we're working with, that we really are, I'd say, a pretty untapped resource as far as just knowing where things are and knowing what good questions are there to ask, and knowing what we've been doing for 25 years, right, on the lands, and sharing that with researchers has always been pretty satisfying and has been a good contribution to them. All right, so the next set, we're going to talk about some invasive plant research questions. As I said, invasive plant stuff is so hugely important to what we do. So we just got a couple here. These are both really interesting ones, though, I think, for all of us. So the first one is talking about seed biology. So seed biology characteristics, they're just super, super critical in terms of informing our management strategy, right? So a couple real basic examples of that, right, are like if you have a grass where the seeds only last 1 year, we've got a couple of species like this. That means that if you can, you go out and you're trying to eradicate this population of invasive grass, you spray it, you kill all the adult individuals, things are going to be germinating from the seed bank. If we know the seeds are only good for 1 year, that's honestly just great, right? Because we can monitor it for a year, and as long as we let no plants get to reproductive age within that year, we can pretty much say that that population is controlled, is gone, eradicated. Contrast that with something like Miconia, right? Unfortunately, we know like the Miconia seed bank now, I think, is above 15 years, right? So if there's a mature Miconia that's found, it means you have to go back to that site for at least 15 years before you can actually say that that spot has been eradicated. So, and that's just talking about seed viability, right? There's all kinds of things that come into the seed biology of these weeds that are interesting and important in terms of control. There's germination ecology, there's how long they do persist in the soil. Like we were saying, there's dormancy, basic different kinds of dormancy are going to impact how it behaves and what happens to the population. There's movement of the seeds, right, in the landscape. Miconia is spread by birds. All different sorts of dispersal occurs. That's an important factor in understanding how these weeds are performing and invading. And then there's also ways that we can control seeds. There's pre-emergent herbicides that will kill the seeds when they germinate in the soil. And so that's an interesting tool, and understanding how those could be used to do weed control and eradicate incipient populations is an variable. So that's just a quick little, that in a nutshell, seed biology and weed control. Herbicide efficacy. So this is one, interim for many years. James Leary worked with you guys at UH, and he was a huge cooperator with us. We did all sorts of great trials, he and Jane mostly in the field, doing all kinds of herbicide treatments on IPA, which is that incision point application, using highly concentrated doses of various chemistries on many, many species to understand what methods of control were available, and really improved our efficiency and how effective we are at control many of the weeds that we're trying to control in our management units. He also developed that herbicide ballistic technology, I'm sure that everybody knows about, which is another really interesting way to consider delivering herbicide to various weed species, and is a huge benefit in some environments. So yeah, he's gone now, and unfortunately, there isn't somebody right now doing that, but that need still exists. All of this kind of feeds into developing special local needs labels with Department of Agriculture that are required before we're using any of these chemicals for controlling our threats in the wild. It's not a simple process, and it really takes a lot of work, and it takes a lot of effort. There's also things like organic herbicides, right, that are out there. Everybody is very anti-Roundup these days. There's lots and lots of interest in organics. And honestly, we would love it if there was stuff that we could use that was not a pesticide or a harmful chemical out in the environment. If there are ways to control things in other ways, we'd definitely be interested in that. Grasses are a huge problem for us, and grass control is something that we don't have very many tools to combat. And there probably are many other tools out there that could be considered for that single threat.
Yeah, I think Joby covered that pretty well. I think these two questions are just kind of a start, but like, how do we improve our weed control, which is one part of like a restoration process, right? So how do we get rid of these invasive plants, and how do we understand them better? Because that'll help us get rid of them better. And then what tools can we use for this type of, to actually remove the plants. And then there's tons of other potential restoration questions that also kind of could play into all of this that aren't up here, but like we've been doing restoration for a while. So, how successful are those efforts? What species are the best to put out? I do see one question in the chat. Is the study restricted to Army controlled land? So I guess short answer would be no. Definitely on the Army lands, on the training ranges, it's very easy for us because we can just work directly with you on a study. So if you did want to do a trial of something or a study of something and it involved working on, say, state land, then you would we would be happy to work with you. But if you would want, if part of the trial was on state land, then you would have to also talk with DLNR and get their approval and permission and potentially a permit, depending on what you were wanting to do.
It's probably also worth mentioning that for the RFP, which went out, because that is funded under this directive, if it doesn't benefit the ANRPO managed lands, it's unlikely to be selected for funding.
Do you have interest in any fire-related projects with fire ecology, post-fire management, that kind of thing?
Yeah, that's a great question. We actually didn't include fire questions on this, but we've been talking with Dr. Daler a little bit about some potential fire-related projects and native impacts to native plants from fire and interactions with invasive species. So yeah, those fire is an interesting one. There's sometimes the parts of the training range and parts of the state lands are threatened by wildfire, like, kind of, what's the word I'm totally blanking on because this is a presentation and everyone's watching, but vandalism, those types of kind of arson, thank you, intentionally set fires are one thing. And then sometimes we also deal with fires from military training. And it's challenging to know what to do to help break the cycle of climax, guinea grass, and haulicoa, and to break that cycle so we're not as susceptible to fire. So yeah, that we would definitely be open to questions along that line. There was another question on Marine Corps land. Right, so That is actually, we actually don't work, we only work with the Army. We don't work with the Marines. They do have their own environmental office, and we could probably put you in touch with somebody there. They would have their own process and everything else for doing research. And I think in terms of water quality testing, with regards to our work, we don't really have much impact with, that's not kind of one of our management goals of water health and water quality. So we don't have that, as that would be a harder one for us to justify with our spending.
I would just add on the fire that Clay is definitely very interested in fire, and I'm sure will continue to be. So he would be a great person to talk to about fire. All right, to move on to some Oahu Elepaio research questions. We've got a couple. We're really excited to work with this native flycatcher bird. The populations have been doing really well. Our predator control currently, pretty much we just do rat control around them, and that alone has been a really effective conservation tool, and populations have been expanding. So a couple of questions we have is because those populations are expanding, which is a great thing, it's actually resulted in a little bit of conservation conflict for us. Some of the areas where the Elepaio are areas that we'd like to do restoration. And in those areas, there might be big guava trees or other invasive plants that the Elepaio are using to forage in, and also at times to nest in. But ultimately, we'd obviously like to take out those guava trees and make it a beautiful native forest. And hopefully the Elepaio would be even happier with the native forest. But there is going to be a transition there where there won't be big trees, because we're going to cut down some big weeds, and it'll take a while for the natives to come back. So we need to understand kind of what is the impact of that restoration on Elepaio. Do they just move over a little bit and they're fine? Does it impact their fecundity? Does it mean maybe they're not going to nest as successfully? We don't really understand what the impacts of restoration could have on Elepaio. So that's an interesting question that we'd like to get more information on. The second one is another interesting one. You know, I already kind of prefaced this by saying that we've been pretty successful with the rat control in terms of having a good conservation trajectory with the Elepaio. But there's a lot of other things, right, going on with Elepaio. You know, there are many other factors that obviously contribute to their breeding success every year and their longevity in the wild. So what are the other things that maybe we should be keying on? You could think about such factors as we're pretty sure that young birds aren't as good as old birds at raising babies, just because they're new and they have to learn how to be good parents. And so if that really is true, then maybe if there is a choice, we should do more predator control around the more adult birds, because they'll probably be more effective at raising young with the predator control. Whereas the other birds, even if there is predator control, they might not be as efficient at it. Vegetation is another question. You know, I just brought up about guava and native forests. Lots of our Elepaio today exist in completely alien forests, and they seem to be doing pretty good. But could they be doing better if it was native forest? How important are native trees for Elepaio? You know, and that would play into probably a few factors, like is there more food available in a native forest versus an alien forest? What about the predators? You know, are rats in native forests less apt to climb up the trees than they are in an alien forest where there's all this fruit like guava up in the trees? We know they love to climb the guava trees and eat the fruit when it's ripe. It seems like that could be bad for Elepaio. So factors like that, many other obviously factors that you could think about that could play into what makes some Elepaio more fecund than others.
Yeah, I guess both of these questions I think are really interesting, because we've had a lot of success with Elepaio thus far, in that they do great with just predator control. And our rare bird expert on staff has been doing surveys and finding them moving into new territories and areas where we're not doing control. So this question of like, how do we work safely around Elepaio is a bigger and bigger question for a program, because they are expanding, which is a wonderful problem to have.
So our rare arthropods, we got some really interesting rare arthropods we work with. Native damselflies, picture wing, Drosophila. So I'm going to kind of fly through these pretty quick so we can get to the end. So limiting factors, very poorly understood what the limiting factors are, in particular with our Drosophila, our flies. They got complex ecologies. They've got different kinds of threats. They've got threats that obviously eat them when they're adults. They've got problems with competition, maybe even in larval sites. On this number 10, we're still like, we're using monitoring techniques that were developed 50 years ago, right? To understand how many of these flies are out there. You know, Carl Minyak is working with us. He's like awesome, like one of the best fly guys in the state, in the world. And, you know, we just don't know whether or not there's a better way to monitor them. We'd love to know whether or not there was. We use mashed bananas and mushroom juice, right? So that's all about like fermentation and yeast cultures and this sort of stuff. Maybe there's a better bait that can be designed. The bees, in recent years, there's been some great developments with bees on the coast where they're using nesting blocks where they just drill holes in the blocks, and the bees like to nest in these little blocks of wood, and it's great for increasing nesting habitat as well as monitoring. We've tried to move that Malka and it's failed. And we'd love to understand if there was a way that we could develop nesting box technology that would work more Malka in wetter environments. We think it's pretty much a moisture issue. The last one on this one is damselflies. So, you know, we've got the orange-black damselfly and tripler. We're working with Will Haynes, who has this incredible program over at Kauai Nui. He's breeding the flies. We've been releasing them at Dillingham with some really good results. But we had this really big scare this year where both at the wild population in tripler as well as at his breeding facility, we had this massive die-off, big mortality event. We don't know why. It'd be great to understand why that happened, because we don't know if we're going to be able to, like, if it's going to happen again, how would we manage it? We don't have any idea even what it was. Okay, so plants, here's a couple of our beauties, Cyane superba and Stenagine kaneoana. As I said, the plants are the majority of the taxa that we work with. Lots of plant-related research we've done over the years. So let's see. The first one is a very interesting one, looking at beneficial fungal associations. We've done some of this with the Hinson Lab already and had some really interesting results that kind of show that if there's beneficial fungal communities that are established on the plants, they actually can give them some resistance and some vitality in the wild. Now, when we're growing things in our greenhouse, unfortunately, mostly we're going for like sterile, 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 sterile. So all of our stuff is like super clean. Our biggest concern is about introducing new pathogens into the wild. We don't want to do that. But we're really interested in whether or not there would be ways to explore this beneficial fungal associations more. The next one is a massive one. So this next one is soil, right? Like, and I understand a lot of people thinking about soil and NRM these days. That's great. I was talking to Melissa, the other Melissa today, and she's thinking about soil, which is awesome because as managers, 25 years, we've barely done anything with soil. Like we don't know, you know, like soil structure, soil chemistry, soil composition. What do weeds do to soils? You know, you look in guava patches and the soil is definitely different than it looks like in a native forest. What does that mean? What does that mean in terms of our rare plants? We should be looking at soil more. We should be trying to understand what we could do to manage it. We should be looking at how we can monitor and determine where we might have better places to do our rare plant work. That's soil in a nutshell. Okay, let's see, cryo storage. So this is another real interesting thing. It gets into seed lab technologies. So seed lab stuff, seed storage is an incredibly important part of all plant conservation actions. Storing seeds is your insurance policy. It's the way that you hold on to them for a few years until you get your fences built and you have habitat protected. It's a great way to keep hundreds, millions of propagules safe and you know, secure for the future should you need them. But not all seeds store equally. And some of the species that we really need to store, we can't because of their seed characteristics. So Lion Arboretum has developed or has invested in a cryo storage, which is super cold liquid nitrogen storage facility, where we want to look at storing some of these species that we need to store and thus far have had difficulties in our sort of standard storage techniques. All right, and the last one on this is specific to a plant that grows out in Kahuku, a Nioi or Eugenia coelauensis, that is pretty much being extirpated by this Austropuccinia rust. So this is kind of akin to, if you don't know this example, it's like a rod situation. There's a invasive fungal pathogen that comes in and destroys plants. And this has resulted in us pretty much rescuing all of the plants from Kahuku. We've got 159 founders represented in our living collection that lives in our greenhouse. And we really want to take it to the next step and understand how we can address this Ostropyxinia threat in the wild or in a botanical garden or in somewhere else, so that we can move beyond just having these plants in pots. Yeah, so that's, I think I'll make it. I think I got one more, Melissa. Let's get through them. This one. All right, this one's quick. Okay, so endangered tree snail research questions. As you know, we're close to Melissa's heart. Here's Kahuli tree snails, Achatenella mustelina, one of our favorite little buggers that lives up in the mountains. And here's a horrible picture of a Euglandina sucking out a live Achatenella in a tree. So this is one of the major reasons why our snails are as rare as they are. They suffer from being eaten by Jackson chameleons, rats, and then this, the introduced predatory snail, finds them and kills them. So we've developed snail enclosures, which I think most people are pretty familiar with. We work a lot with SEP. Dave Sisko, his crew is awesome, doing snail conservation full-time on Oahu, where we've got lifeboats, more or less. We've got snail enclosures in most of the critical areas in the Waianae Mountains, and we've got our snails in those areas. Pretty much anywhere else outside of those enclosures, the snails are crashing. Within my career, for sure, there'll be probably, given the current trajectory, there'll be no snails left outside of our enclosures. So the maintenance of these enclosures is critically important. One of the hardest things to do is know when the Euglandina are out of them, when we build them and they're fresh and new, and then make sure that the Euglandina stay out of them and don't get in them over the years while they protect our snails. So we've done things like worked with dogs, which is something that Dave Sisko is looking at right now. We're going to be working with another company to try to bring them in and see if we can use dogs to detect them. We've dreamed about eDNA, and there's probably all sorts of other ways that you could figure out whether or not there's an invasive snail inside of your snail enclosure, because we really need a way to certify them annually to say, yep, predator free. So yeah, I think that's it. So yeah, let's open it up for discussion questions on any of those last three that I zipped through.